Hello, my name is Deffge. I'm a medical doctor at University Hospital in Magdeburg. And I'm working in the cardiovascular research group of Dr. Herold and Professor Braun-Dullaeus. We investigate the augmentation of collateral vessel growth. Therefore, we use live cell imaging. In this video, we want to explain our methods.
Resuspend the cells in serum-free culture medium with the density of 1 × 10⁶ cells per milliliter. Add 5 microliters of 1 millimolar DIO in dimethyl formamide to 1 milliliter of the cell suspension and resuspend carefully. Incubate the cell solution at 37 degrees centigrade for 20 minutes. Centrifugate the cells at 500 G for 5 minutes. Dislodge the supernatant and resuspend the cells with 37 degrees centigrade warm FCS supplemented medium. Repeat this step twice. Count the cells with the help of a Neubauer counting chamber and light microscopy. Resuspend the cells with sodium chloride solution. Inject the cells into the tail vein.